Greetings, I am M. Chatya from 2nd MSc Microbiology. In this video, let us discuss about genetics of nitrogen fixation. Nitrogen fixation. In nitrogen fixation, the nitrogen is split up into free N2 atoms by breaking the triple bond with the help of an enzyme, nitrogenase. Nitrogen fixation was observed by Burris in 1966. Nitrogen fixation can take place by two methods, which are non-biological nitrogen fixation, which is also called as physical or natural nitrogen fixation, and biological nitrogen fixation. Non-biological nitrogen fixation. In non-biological nitrogen fixation, nitrogen can be fixed by lightning, converting the nitrogen and oxygen into NOx, that is nitrogen oxides, where it cannot be used by plants. Nitrogen oxides may react with water to make nitrous acid, which seeps into the soil, where it makes nitrate, which is of use to the growing plants. Biological nitrogen fixation. It occurs when atmospheric nitrogen is converted to ammonia by an enzyme called nitrogenase. The overall reaction of biological nitrogen fixation is nitrogen plus 16 ATP plus 8 electron plus 8 hydrogen ions gives 2 ammonia plus hydrogen plus 16 ADP plus 16 phosphate ions. Microorganisms can fix 70% of nitrogen and are called diazotropes. After a small introduction, now let's see what are all the genes and enzymes that are involved in nitrogen fixation. Nitrogenases. Nitrogenases are complex enzymes that contain two components, namely iron protein and molybdenum iron protein. The smaller component known as the iron protein, which is also known as non-heme iron protein or dinitrogen reductase, contains two subunits and having the molecular weight of about 60,000 daltons. The larger component known as molybdenum iron protein or iron molybdenum protein or dinitrogenase contains 4 subunits having the molecular weight of about 2,20,000 to 2,70,000 daltons. The iron protein functions as an ATP dependent electron donor to the molybdenum iron protein, which contains the enzyme catalytic site. Both of the nitrogenase component proteins are extremely oxygen sensitive, that is, active only in the anaerobic condition and are degraded in aerobic condition by oxygen. There are three major genes that are responsible for nitrogen fixation, and one of them is NIF genes. The NIF genes fall into three categories, namely structural NIF gene, regulatory NIF gene and supplementary NIF gene. There are 20 NIF genes that are involved in nitrogen fixation. The iron protein is encoded by NIF-H gene and the molybdenum iron protein is encoded by NIF-D and NIF-K genes. The NIF-D, H and K genes are recognized as structural NIF genes and they are responsible for the control of nitrogenase enzyme complex. The second notable gene is node genes and they are located near NIF genes. The nodule forming genes are present in plasmids in case of symbiotic bacteria. Totally 10 node genes, of which 4 are responsible for nodulation and remaining are host specific. Node genes control the activity of two phytoactivators that are indole acetic acid and cytokinin. They are 8.5 kb in length. The third notable gene is HUP genes. Molecular hydrogen can be utilized by a variety of bacteria which have evolved hydrogen uptake. The HUP genes play an important role in plant productivity by recycling the considerable quantities of hydrogen which is wasted during symbiotic nitrogen fixation. About 30 to 50% of hydrogen loss has been recovered. It helps in formation and maturation of hydrogenase enzyme. Regulation of nitrogen fixation by NEF, node and HUP genes. These genes collectively enable nitrogen fixing bacteria to efficiently convert atmospheric nitrogen into ammonia. They are essential for the ecological and agricultural significance of nitrogen fixation as they contribute to the fertility of soils and the growth of nitrogen-demanding crops. The regulation of NEF, NOD and HUP genes is interconnected to ensure that nitrogen fixation occurs efficiently and under appropriate environmental conditions. Environmental signals such as oxygen levels, nitrogen availability and plant signals play crucial roles in modulating the expression of these genes. This regulation ensures that nitrogen-fixing bacteria can adapt to varying environmental conditions while maintaining effective symbiotic relationships with plants. Thank you for watching.